I would like to introduce Professor Dimitris Thanos, who is a member of the Academy of Athens, and he is one of the key contributors to the FirstSteps program for the last few years. So I don't think he needs a lot of introduction, and he's going to kick off with the first plenary talk. Dimitri?
Okay, thank you very much. My name is Dimitris Thanos, and I'm the Scientific Director of the Biomedical Research Foundation of the Academy of Athens. And today, I'm going to try to combine two big genomic initiatives which are active in Greece. The first is the Genome of Greece, and then the Genome of Greece meets the newborn screening. The newborn genomic screening. So population genomics is where human health meets precision medicine. It provides profound insights, as you see there, into health, into human origins, evolution, and also migration history. And this is a very important component. So why genomics matters for Greece? Of course, besides the obvious medical reasons, which I will not describe in detail, the thing is that Greece has become during history a living genetic laboratory, and this is because because Greece geographically is at the crossroads of migration. It has a very rich history, and also has a really long history on isolated communities, and our genetic continuity with everything I said makes Greece actually a living laboratory. So before a genetic laboratory, a real genetic laboratory. So this is only to remind you that all of us in this room, we differ only at 0.1% in our genome, and these differences are enough to make us different the way we look, but also plays a very critical role in how we behave, and of course provides all the predisposition to diseases or make us sick. However, this has to be taken into with very big caution, because the return of the genomics back in the public domain also can create race-based thinking. So I remind you, to all of you, that genomics has already revealed that human genetic diversity exists on a continuum. So you cannot really look at the genome of someone and saying that he is Greek, or he's black, or he's whatever he is. So the very important thing is how to apply genomics. You know, Greece, the last 10 years has really has been serious in building genomics, and to do that, you need 5 different things. You need sequencing labs. You need people to analyze and handle the data. So you need bioinformatics, secure storage of the data, and of course accredited clinical labs to actually verify and perform all the clinical genetics work, and of course a trained workforce. And I think Greece has very slowly but very effectively has started actually to build all this infrastructure. Now, the very important thing is that these infrastructures, the way I see it, begin with the physical and human infrastructures, move to the Genome of Greece, and I'm going to talk a little bit about that. then to FirstSteps, the initiative for the newborn genomic screening, and of course at the end, we actually reach at the point of clinical clinical translation. Now, regarding the infrastructures, 10 years ago, we built the first and still only genome center, which is called Greek Genome Center, which is fully equipped with all the fancy machines you can imagine, which is constantly upgraded. So the big project which I'm going to start talking first is the Genome of Greece, which is actually to generate a genome, a genome version, or a version of the genome, depends how you look at it, which actually represents the Greek population. And the reason for doing this is very obvious, is to translate genomic and health data into scientific breakthroughs, to help people to get a better treatment and diagnostics, and of course, one of the very important components, because we Greeks are very sensitive to history and to the past, is actually to reconstitute the ancient history of the country. Now, the Genome of Greece is part of the big European project, which is called Genome of Europe, and I see Stylianos and Barta, who are actually in the board, in the scientific advisory board of the project. So Greece will, at the end, will sequence 36 genomes, 36,000 genomes. 18,000 are healthy, or they think they are healthy, and 18,000 disease genomes with all the known disease we have. Now, for the moment, we have begun the first phase, which is about 3,000 genomes. Now, the genome of Greece, as I said, is going to be very critical in actually provide these national allele frequency maps. I mean, what are the alleles throughout the country, the haplotype blocks, and everything else we need to actually implement a clinical medicine in Greece. We need to know what kind of pathogenic variants are in the country. And I'm going to show you some preliminary results from the newborn sequencing project regarding this. As I said, Greece is a tapestry woven. So it's a mixture of different, are remnants of different populations which have been migrated in the country. We have different haplotypes in different regions. I don't have the time to actually discuss those. And of course, we have islands, isolated islands, and we have islands like, for example, Ikaria, where the life expectancy is about 10 years more than on the average in the rest of the country. So we really want to look at this, and in fact, I remind you, about a month ago, there was a publication, I think in Nature or in Science, I can't remember, where they claim that 50% of the longevity in humans is due to genes, and the other 50% is other reasons. And of course, the studies in isolated regions have led to the identification of very, very rare diseases because of the endogamy in this very isolated space. For example, in the Naxos Island, the Naxos disease, which is a placoglobulin, the gene of placoglobulin, which actually cause heart diseases and also dermal disease. Now, The thing is that we live in a really very nice area and a very nice climate, and it is for sure that our environment or our genes have been adapted to this environment, and we really want to know how. And this is one of the long-term goals of this project. And of course, Greece have gone in many, many different places, and about 3% of our 3 to 4% of our goal is actually to sequence Greeks which live abroad. Now, this is the distribution of the population in Greece. You see that. You see, of course, in the Attica regions, or nearly 1/3 of the people live here. So we have biobanks which actually represent the population of Greece. You see those which are at BRFAA, were stored at BRFAA. And in fact, we will focus, and this is my personal interest, scientific, of course everybody else in the group has its own interest, is actually focused on isolated islands and mountainous regions and in Mount Athos. Now in monks living in Mount Athos, and we have a biobank on that in collaboration with a Greek working in France, where we will sequence, these people live in a very stable environment, have a very controlled environment, not stable, very controlled environment. So we are really very anxious actually to find out how this interacts with their genome. Now, this is a Genome of Greece. We think is going to provide the base, the foundation for actually move to the newborn sequencing. As Petro said before, this is a public health issue, which I will not repeat the trivial things for early detection, personalized care, prevention. This is very important, and of course research, cost savings for the national health system, and of course equity, which is a remarkably important issue because all children have to be found, as Barta told me 100 times. Every child has the right to be found, right? Right. So it has to apply to everybody. Now, what is the purpose? The basic purpose is actually to identify the children which have a treatable disease. We know that about 1 out of 20, 5% of the children are being born with a genetic disease. Takes about 5 years on the average to be diagnosed, and needs 7 doctor specialists to be found. Now, 50% of the children which die at the hospital in infancy, 50% of the children, this death basically is due to a genetic disease which hasn't been identified. Finally, I would like to finish with the last 2 slides. The implementation of FirstSteps, which is the newborn sequencing program, will increase the diagnostic yield, and of course will provide earlier diagnosis for therapy whenever this therapy exists. In fact, there are about 500 different diseases which can be therapeutically approached, genetic diseases. So there are many such programs in the world. You see many of those, and I'm going to finish with this. We completed phase 1 with 417 children which have been sequenced. 4.5 were turned to be positive, and many of the data have been verified by Sanger sequencing, and also we have sequenced trios with their parents. What we found was 8 confirmed diseases, so the children have symptoms, are sick. And of course, we are waiting for 9 lab result validation, whereas other require further analysis. And we have identified in these 417 kids, we have identified 255 pathogenic or likely pathogenic variants, which is a number which sounds very scary, but it is the truth everywhere, not only in Greece. We are not sick by default. So Genome of Greece will help FirstSteps because it is the critical interface will provide, I'm done. It will provide the critical interface between population genomics and clinical implementation. The ethical stuff will be presented by Barta, so I will not repeat what most likely she will say. So I leave it to the experts. And I'm going to finish you with this. I found this is remarkable. I would like, first of all, to thank all of my colleagues at the Biomedical Research Foundation, and IVE, as we call it in Greek, and Petros here for the FirstSteps and all the people who have been involved. And I'm going to leave you with this. This is a saying from Frank Gannon. Frank Gannon was the EMBO director for 8 years. When I was elected EMBO member in 2004, Frank Gannon was the EMBO director. So he said that science is a reliable repository of truth, which is an amazing expression. And I think the DNA sequence is a repository of truth. Thank you.